Welcome back, guys. In this video tutorial we'll be talking about the enzyme linked immunosorbent assay, or simply known as ELISA. ELISA is one of the very much sensitive technique to detect the presence of antigen or antibody in blood, right? So that's why we use ELISA. This technique is kind of similar to that of RIA, or radial immunoassay. In this radio immunoassay, what we do actually, we use radioisotopes to see the presence of antigen or antibody in the sample. But in this case of ELISA, we use enzyme substrate reaction, and the coloration due to the enzyme substrate reaction as a process for identifying the presence of antigen or antibody. Now, the major important thing about ELISA is that it is extremely powerful and extremely sensitive, because ELISA can give us the idea of or identification of proteins in picogram level. So that's why really important for all the process. So let's talk about the ELISA as an overview. So what we do in ELISA, let's say you have an infection in your body. In any case, if you have any infection, that means due to any bacteria or fungi or virus, in either cases, our body produces antibody against those infectious agents. Now those infectious agents and their materials that is inside our body are termed as antigen, and to fight against those antigen, our body produces antibody which are specific towards that antigen. Though it's required more time for our body to produce more antibodies against the antigen, but it produces it antibody in higher amount after some time of the infection. So if you find certain types of antibody in your body, that means you have the infection of a particular type. So if we can detect that presence of a particular antibody, we can be sure that the infection is occurring. So that's why ELISA comes in to give us the idea what kind of antibody is present and what kind of antigen is present in our blood, in our serum. Okay. So if we see here, it actually it's a assay, it's a technique that's called ELISA assay or ELISA technique, whatever you say. It contains particular some type of glassware sets like this one, as you can see here. This is called as the micro titer plate. This micro titer plate is nothing but a plate containing small wells, as you can see here, the well like structure. Now these wells are, there are different types of structure. Microtidal plates are available. Some of them contain 96 wells, some of them even more wells. Usually they contain 96 wells. So 96 small wells are present here all around this. So what we do now, we add the sample at the first in this particular wells. Now the thing is, this is not just a well containing slide. It has certain other very important properties too. For example, if we zoom into one of these wells, if what we can see here, there might be different structure, but generally if I zoom into a well, it will look something like this. If you look from the longitudinal section, you can see this. These wells are covered with specific type of antibodies. Remember? So let's say these are the antibodies, the Y-shaped structure. They are added, they are attached to the well bottom. So at the bottom of the well, all these antibodies are attached, and their FAB portions are free to react. Now in this type of wells, what we can do, we can add sample now, because our sample may contain antigen. If our sample contains antigen of a particular infection, we can diagnose that particular disease. So let's add that antigen. Here the antigens are depicted with this triangular shaped molecules. So once we put the antigen in the place, those antigen will specifically interact with the antibodies that are adhered to the bottom of the wells. Once they adhere there, what we do a washing stage each time, because for the unbound antigens to be washed away. After that, the second and most important part of the reaction is to add another type of antibody which are again specific to those antigens, but this time the antibody contains an enzyme linked with it. For example, if you see here, these are again red Y-shaped antibodies, but they are attached with some kind of circular red dots, and those circular dots here nothing but enzymes. Now these enzymes can interact with particular type of substrate to convert that substrate into a colored product which we can visualize. So now if you see here, Now this antibody along with those enzyme linked, they will go and attach to the antigen, right? And then we add substrate here is it's yellow in color, and they'll give some coloration. Due to the coloration we can see the presence of the antigen in those wells. So this is the general process of the this is the idea that we use the antigen antibody interaction, and we use this antigen antibody interaction to find out whether the antigen is present in our serum or whether antibody is present in our serum. So there are different types of ELISA. For example, if we look at here, this is the first kind we will be talking about is the indirect ELISA. There is also a thing called as direct ELISA. The previous model that we've seen here is called as the previous model. Remember, this one is called as the sandwich ELISA. Why, we'll be talking it later. But in the indirect ELISA, what happens? Let's say this is the well. Whatever we're looking now, right now are the structure of the wells, right? We're not looking at the overall structure of the microtiter plate. But if this is the well, and you see here red dots are present there, so we add the sample into that well. So sample is present into the well. Now the second stage of reaction, what is to add the antibodies. So we add the sample, sorry, actually, okay. Okay, so let's talk about indirect ELISA. In the indirect ELISA, let's see, this is the well. So whatever we're looking at, the structure is the structure of the wells. We're not looking at the complete structure of the microtiter. We're instead looking at a well in the longitudinal section. So if you see here, this well is now covered with some antigens. Now this is completely manufactured by the laboratories and companies. So you can see here these red things, though they are few, but actually in the wells there are many more in actual reality. So those antigens are coated, and they are very specific to adhere to a particular type of antibody, right? So now let's say upon a particular infection in our body, we develop certain antibodies. Now we add the serum sample to this well. Once we add the serum sample to the well, the antibody can bind with this antigen because of the specific antigen antibody reaction. So after this specific antigen antibody reaction, as you can see that those antibodies from our serum, from the sample, is now attached with the antigens that are adhered to the bottom of that well. After that we add the secondary antibody. Now the secondary antibody is very important, because the secondary antibody carries the enzyme. Now the secondary antibodies are not like the primary antibody that we have added, because in the secondary antibody what we can see is that they are attached with an enzyme, they are linked with an enzyme. That's why it's called enzyme linked immunosorbent assay, right? Enzyme linked immunoassay. So for that reason, as you can see here, that enzyme is linked, and after that we will add our substrate, because the enzyme definitely will interact with the substrate to convert it into product. So we add the substrate, and due to the reaction of this enzyme with the substrate, it can convert that substrate into a colored product. So the color of the well, color of the solution that is present in the well changes, and we can detect the presence of that antibody. If this whole process at the end will not result in any coloration in the well, that means there is no antibody present for that antigen that we test for. Now the thing is, in the laboratory we can create different types of antigens, we can add different varieties of antigen in different wells to produce different specific reaction kits, right? Now this process is called indirect ELISA, because in this case the first antibody, the primary antibody that we use, is not enzyme linked. The secondary antibody is enzyme linked, and that is the anti-primary antibody to react with. So this process of enzyme linked assay is not direct. That means the first antibody that we use is not attached to the enzyme. If the first antibody that we use is attached to the enzyme and it will give us the result, in that case it will be termed as the direct ELISA. But as this process continues with two different antibodies and the second one is enzyme linked, it is termed as indirect ELISA, because the process of enzyme substrate reaction occurs via the primary antibody. That's why it's called indirect ELISA. Now let's look at another ELISA process, and this is called sandwich ELISA. Sandwich ELISA was taken to determine the presence of antigen in the bloodstream. So if you look at here again, this is the structure of a well. Now this time the structures of well will contain some antibodies adhered to it instead of antigens. So now we add the sample, the serum. It may contain the antigen of a particular disease or may not. Let's say it contains the antigen in red in color. So now the antigen is very specifically bound with those antibodies that are present at the bottom of this well. So after this attachment, the second stage is again addition of the enzyme linked molecule. Again, obviously, as there is an antigen, so definitely to interact with this antigen, we require another specific antigen specific antibody, which will be linked to enzyme. And that's what we add here, the second antibody, not actually in this case the primary antibody, because already the first antibody is adhered to the well, so we don't need to add that. So this is the antibody with enzyme is added. Now we'll add the substrate. Enzyme substrate reaction will take place. It will turn the color yellow again here. After that we can know that yes, the sample that we used definitely contains the antigen that we look for. So that's how we can find out the presence of an antigen in this sandwich ELISA. But in the previous case of indirect ELISA, we know the presence of antibodies. Now let's look at the third type, which is called as the competitive ELISA. This is a slightly different. Actually, if we don't know that which is the particular antigen that is causing a problem or what is the particular antibody that is causing a problem, then we use competitive ELISA to figure out which antigen is causing more problems and which is not. So what we do here in this case, in competitive ELISA, we take the antibody from the patient. Let's say these are the antibody. So what we know is that this competitive ELISA, there can be an antigen attached with antibody, that means the antigen antibody complex, or there sometimes only antibody can be present without any antigen, as you can see in both these cases. Then we add them into the plate. So as we add them inside, what we can see here, this wells that I haven't mentioned, that the thing is the wells that we look for this competitive ELISA, they already contain these antigens, just like the indirect ELISA wells, they also contain antigens. Now remember the previous stage. So actually we should draw something here. So let me draw it because there is a, it should be drawn here. So these are some of the antigens that are present at the bottom of these wells. Okay, antigens are present at the bottom. So this is the well looks like in competitive ELISA. But now when we add the serum, it may contain antigen antibody complex like this left hand side one, or the antibody itself. So once we add those inside, then those antibody which are bound with antigen previously are not free to interact with the antigens that are present at the bottom of the well, so they will just float around. But the antibody which was free previously will be able to attach to the antigens that are adhered to the bottom of the well. So once this process is done, we go through a wash tape and then we add the secondary antibody here, which is linked with which is not actually secondary here, it's enzyme linked antibody. Here when we add this enzyme linked antibody, which is also specific, then it can bind with the Fc portion of the first antibody that was from the sample. Now then we add the substrate, and the enzyme substrate reaction we turn the coloration of that well, and we can find the presence of that antibody. So this is the idea of competitive ELISA. We can detect whether the antigen antibody complex is present or only the antibody is present. Though we can also use the complement fixation assay to prove that, but also competitive ELISA can give us the idea. Now the best thing about ELISA is that it is not only qualitative. As you can see, we can detect the presence of antigen, we can detect the presence of antibody. But the best thing about ELISA is that it is quantitative in nature. That means we can actually tell how much antibody is present in the sample, how much antigen is present in the sample for a particular disease. And that is the most important point. So what we can do here, if you look at the wells of ELISA, if we go and start looking at the wells of ELISA, you can see in these wells, for example, in these wells, what we know about that, throughout the well, what we do is a twofold dilution from one end to another end. So once we pass through this twofold dilution, that means from the first to the last well, there will be twofold dilution all over the place. That, and what is twofold dilution will be talking in the other videos. But the thing is, we are diluting the sample, whatever sample we take, whether it's an antigen sample or antibody sample. Generally, it's a serum, a patient blood serum sample. It is continuously diluted. So as we are diluting this sample throughout this wells, we can actually detect the percentage of antigen or antibody present in each different wells, right? That's why we can quantitatively manage that, quantitatively understand that, and we can also tell the exact concentration in the picogram level. We can construct a graph with these values, and that's why ELISA is very important, guys. And I hope that's helpful. If you like this video, please subscribe, vote for my video, like my video, share my video in social networks, and thank you very much.